Hi, good evening, everyone.
Hi, good evening.
I would like to welcome Dr. Bhawika Lamichhane. She's a biomedical researcher at the University of Utah School of Medicine. She currently studies genetic processes that regulate blood cell development and innate immune cell behavior. She's passionate about science communication, and that's why she runs and manages our KAT6A, KAT6B registry. And
I don't know how.
A portion of her talk will be about the registry.
Hi. Hi, everybody. Thank you for coming. Today, the title of my talk is Understanding Rare Diseases, KAT6A, KAT6B, and the Patient Registry. First of all, I want to acknowledge Rare Disease Month and Rare Disease Day. Rare Disease Month is observed in February, and Rare Disease Day is going to be celebrated on February 28th, where patients from all around the world will be celebrating this special day. So when we look at rare diseases is defined as a condition that affects fewer than 200,000 people in the US. The term rare disease was coined by US Congress in 1983. The number of people who have rare disease encompass 0.06% of the US population, and the need to acknowledge or give a term rare disease was driven by the acknowledgement that drug companies were not interested in developing treatments for these diseases that have very few patients. When we look at worldwide stats, there are currently 7,000 rare diseases suffered by patients all across the world. 300 million patients worldwide have some sort of rare disease, and this encompasses 0.04% of the world population. Majority of the rare diseases, as we know of, are thought to be genetic, mostly de novo mutations, meaning these mutations are freshly acquired by the patients who have the disease. These mutations are not inherited from their parents, but in some cases, a small fraction of rare disease are inherited. So before I walk you through KAT6A and KAT6B, I wanted to kind of touch upon mutations and why do mutations create problems. So as I said before, most rare diseases are caused by mutations. And so our DNA sequence that we have, the DNA sequence of a gene is read in 3-letter code called the genetic code. And so 3 letters of DNA corresponds to an amino acid, and sequence of amino acid is organized to form what we call protein. So for example, when we look at the KAT6A gene, it is made up of 612 letters or nucleotide bases, and the KAT6A protein has 2004 amino acids, so 6,012 divided by 3, which is 2004. So any changes in the DNA sequence, or which is a mutation, can alter this 3-letter code and make faulty proteins. So let me give you an example. Let's assume this is a DNA sequence, this string of letters. So if we read this string of letters with the 3-letter code, what this translates to in a protein form is the cat ate the big rat. Now, let's assume there is a deletion mutation where a couple of the letters or bases are missing. And so even with a couple of letters missing, now this sequence of letters, instead of translating to the cat ate the big rat, is going to translate to the cat ETB, ETHEBI, GRET, nonsense. So just 2-letter introduction completely changes how this sequence is translated. Another example of a mutation is an insertion mutation where let's assume just one new base or one new letter is being added to the sequence. And when we look at that, now this particular sequence is translated to, instead of the cat ate the big rat, is now translated to the cat had ETHEBI, rat, nonsense. Similarly, a third kind of mutation, a substitution mutation, where one of the letters is being substituted by a different letter, can also completely change the meaning of the sentence, the protein outcome. So as you can see, because all of the protein that is being made is followed by a 3-letter DNA code, even a single base alteration can cause devastating consequences in terms of the protein that's being made. So with that, I want to introduce you to the KAT6 genes. So the KAT6 genes function in our body by helping unravel the DNA. So in order to fit all of the DNA that we have inside of the cell, the DNA is tightly packed as chromosomes. And so with the help of these proteins called histones. And so as you can imagine, not all of the DNA sequence, all of the genes are open and available for gene expression at all times. So there are genes and proteins that we call epigenetic modifiers that act in controlling what part of the DNA is open versus closed, depending on the need. And so KAT6A, both KAT6A and KAT6B genes are histone acetyltransferases. They are epigenetic modulators, and they function by transferring acetyl group to histones. And transferring of acetyl group to histone, what it does is it opens up the DNA, and this allows for the genes to be turned on and allows for gene expression. Besides making DNA, our genes available for expression, KAT6 genes also switch important proteins from inactive to active state through a process called post-translational modification. And so as you can imagine, with this very important function of making sure our DNA, our genes are open for expression, for transcription, as well as controlling the function of important proteins, the KAT6 genes serve a critical role in facilitating many cellular events in our body. And so we have now known that both KAT6A and KAT6B genes are extremely important in early development, and early development is a dynamic process, and many events have to happen simultaneously and in a controlled fashion, and both these genes are very critical in making sure the right genes are open and accessible at the right time. And so mutations in these genes thus create many problems, as we are all aware. When we look at the KAT6A gene, KAT6A gene is present in chromosome 8 in humans. So far, the known cases worldwide counts up to 350 patients. And mutations of KAT6A gene has been reported throughout the gene. So there are patients who have mutations towards the beginning of the KAT6A gene, all the way towards like at the end of the gene. So throughout the whole KAT6A gene. So the interesting thing about KAT6A gene mutations and many epigenetic modifier gene mutation is that patients present with variable phenotype and variable penetrance of the same phenotype. So different patients can have different phenotypes, and some patients who might have the same problem or the same phenotype can present it in various severity. What's interesting about the KAT6A and also KAT6B gene mutations is that the late truncating mutations, mutations towards the end of the gene result in more severe phenotype compared to mutation towards more early in the gene. Some of the core features of KAT6A gene mutations include neurodevelopmental defects, gastrointestinal defects, cardiac anomalies, muscular defect, immune defect, and sleep disorders. So some of the more severe phenotypes, microcephaly, intellectual disability, gastrointestinal, cardiac anomalies, these are associated also with the more late truncating mutations, the mutations that appear towards the end of the gene compared to early appearing mutations. When we look at the KAT6B gene, KAT6B gene also falls in the family of histone acetyltransferase, same as KAT6A. It lies in chromosome 10 in humans. Here you can see the map of KAT6B gene that I've shown here. So far, the total known cases of KAT6B comes to 100 patients throughout the world. This is the tally from the available literature. And similar to KAT6A, mutations have been reported throughout most of the genes. So from the beginning to all the way towards the end. And also similar to KAT6A, there is variation in phenotype, occurrence, and also penetrance depending on the patient. And the most severe mutations, the most severe phenotypes in patients are seen in patients that have mutation in the last exon, the distal end of the KAT6B protein. Some of the core features of the KAT6B gene mutation is distinct craniofacial features, neurodevelopmental defects, including microcephaly, intellectual disability, global developmental delay, genitourinary and renal abnormalities, cardiovascular anomalies, and musculoskeletal defects. KAT6B patients can actually be differentiated into two distinct groups depending on the phenotypes they show. And so the first one is the genitopatellar syndrome, and the second one being the Otto syndrome or the SBBY-SS syndrome. And so between these two syndromes, patients who have the GPS tend to have much more severe phenotype compared to patients with SBBY-SS. And once again, the more severe GPS syndrome is associated with patients that have mutations in a particular place in exon 18. So towards the end, meaning more problems. And if you look through the phenotypes, the GPS patients do present with much more severe phenotype, like agenesis of corpus callosum, lack of corpus callosum, a really important part of the brain. And so with that, I want to walk you through the KAT6A, KAT6B patient registry. So why do we need the registry? What is the registry for all of that? So KAT6A patient registry was launched in 2019 by the KAT6A Foundation. It's hosted by National Organization for Rare Diseases, NORD, and NORD hosts more than 300 patient organizations. What the beauty about the patient registry, it's hosted in a secure cloud-based platform. So the registry follows strict government guidelines for the management and protection of patient information. Patients can feel good that their data is well protected and follows all of the government guidelines. So it's a very secure, great platform to put all the important patient data. So why do we need the registry in the first place? We're dealing with rare diseases, so any data that we can get is important and useful for us to better understand the manifestations and the nuances of diseases caused by KAT6A and KAT6B mutations. So KAT6A patient registry serves as a database to enhance understanding of full range of KAT6 related disorders. It also allows us to disseminate information quickly and securely to researchers and clinicians. Having a place where the data is well kept allows for better communication, and this facilitates research collaborations and helps us expand and get to the next step of understanding of diseases caused by these mutations. And thus, the registry database can help guide the development of standards of care by facilitating research. The current patient registry layout, and some of you who have filled out the registry are familiar that the current registry has 5 surveys. So the participant profile, you fill in personal information, socio-demographic information, information about education, race, sex, age, age during diagnosis, etc. The medical and diagnostic data, covers details on diagnosis, medical history. The treatment and review system captures data on medication and supplement details, route of administration, surgeries, disease progression, the quality of life surveys, pediatric as well as adult. There is adult quality of life survey for adult patient. And this captures information on physical function, mental health, peer relationships, therapies, etc. And the final survey, the KAT6A survey, captures mutation details, KAT6B specific, KAT6 specific medical details, such as neurological issues, respiratory issues, immune issues, gastrointestinal issues, sleep disorders, hospitalizations, etc. So now that I've shown you the registry layout, you must be wondering, what about KAT6B? And so this is what's being changed, a major change in the registry. We are now changing, we're in the process of changing the name of the registry from KAT6A patient registry to KAT6A, KAT6B patient registry to be fully inclusive of KAT6B patients that are already part of the registry, and hopefully to welcome more KAT6B patients to put in information, fill out the registry so we can start understanding KAT6B disorders better and analyze data and just advanced research for both KAT6A and KAT6B disorders. And so we are currently working with NORD to change the registry layout for just the purpose that I described. And so the new patient registry, first of all, most important, is more inclusive, will be inclusive of both KAT6A and KAT6B patients. The registry, instead of having 5 relatively long surveys, will have 12 much shorter surveys organized in this fashion right here. And we're hoping it goes live in the next couple of months. We've been working with NORD for a while, and we're getting towards the end of the process, and we're quite excited to be able to launch this live. And for patients who have already filled out the patient registry, the interface stays the same. For the families who have already filled out the registry, and for the new patients, this is what the new registry will look like. When we look at the current participant enrollment in the registry, it is encouraging in some parts and not so much in the other parts. So I'm sharing this just to encourage any of you who are attending or listening to please fill out the patient registry if you haven't already. And for the ones who have filled it out, thank you. So out of the 350 known cases, we have seen 231 patients are already registered as respondent in the registry. This accounts for 66% of the known KAT6A patients. So this is very encouraging. Where we do need to make a huge progress is the number of patients who have completed the surveys. And so without the completion, this is where all the data lies. So I really encourage you to start filling out the surveys so you can put in your part of the story, per se, your journey through KAT6A that can be incorporated in increasing our knowledge of how KAT6A and KAT6B affects patients. I also encourage KAT6B patients to enroll in the registry as well. And with that, I would like to share with you future direction. So we want to continue increasing patient participation in the KAT6A, KAT6B patient registry to gather more data, to be able to get more information. We want to continue analyzing patient registry data to learn more about the KAT6 disorders and identify new trends. We have already started doing this, and those of you who have seen some of my Facebook posts, we can now start looking more nuanced. We can start getting more nuanced answers to some of the questions, like look into most common GI disorders and see what the distribution within our patient is. For example, when we look at neurodevelopmental disorders, we can see this distribution of what percentage of patients suffer through what kind of neurodevelopmental disorder. Questions like what percentage of patients spend time in NICU, percentage of patients have high pain tolerance. So these are some of the data that have been analyzed, and some of them have been shared, and we want to continue doing that in the future. and hopefully also utilize KAT6B specific data in order to be able to parse apart, like I said, more nuanced understanding of what is happening inside KAT6B disorders. We want to continue sharing findings to patient families, and my big goal and what I foresee for the foundation is eventually be able to publish the registry data findings in a scientific journal to be able to advance science and research collaborations to increase the understanding of science, as well as invite more collaborations. And with that, I would like to encourage you once again to take your time to fill out the patient registry. And I thank you for your time.